Hello everyone, my name is Hyojung Paik. Today I'm going to talk about our research topic titled Supercomputing-aided analysis of docking property between SARS-CoV-2 and host variation of ACE2 from a large cohort. This work was supported by research program of KST, and this work is also supported by National Supercomputing Center with supercomputing research, including technical support of KSC. And as you know, this research also been conducted using the UK Biobank research under the application number 69112. And I'm Hyojung Paik, and these are co-authors, and corresponding author is Sang-jae Seo from KST. Here's the motivation and goal. As you know, the global pandemic due to SARS-CoV-2 entitled COVID-19 by WHO is still ongoing. As of January 9 at 2021, over 200 million of infections cases were reported, with over 5 million of fatal outcomes by COVID-19. However, the understanding of the interaction of SARS-CoV-2 to human cells, including physical docking property by the host's genetic diversity, is still lacking. So we investigate the impact of host genes, host genetic diversity for the entry of SARS-CoV-2. Simply, we performed genetic data analysis covering 200,000 of individuals in whole exome levels from the UK Biobank data. And in addition, we conducted molecular dynamics MD simulation to scrutinize the relationship between genetic variants of ACE2 and COVID-19 infections using the supercomputing system called Neurion. We use our supercomputing infra. This is the fifth supercomputer of KST, and here is the performance of calculation speed, and it is 40 times faster than the first supercomputer of KST called Tachyon. Here's the result 1. So the table 1 shows the demography of UK Biobank data. Actual UK Biobank data covers 500,000 individuals. Out of them, 200,000 people has a matched WG whole exome-seq data. We use these data and examined whether these populations will represent normal adult populations in the UK using these demographies. And out of 2 million of diagnosis record, and finally concluded these populations represent normal adult population in the UK. And so based on this research, we analyzed VEP to assess the functional impact of genetic variation of ACE2. Here's the overview of the health-related outcome. It means a primary diagnostic trend of individuals in the UK Biobank data. And bottom bar is VP-based assessed functional impact of genetic diversity of ACE2. As you can expect, the non-functional variations shows a higher frequency than the functionally related variation is rarely detected. So, and in the second step, we estimated the free energy changes to investigate the molecular basis for the effects of ACE2 variation on SARS-CoV-2 binding. And we found several meaningful variants, and yellow or highlighted one variant shows a higher energy to bind the ACE2 and SARS-CoV-2. It means this person who has these variants has hard to bind with SARS-CoV, and blue highlighted one is easy to bind position, which means individual who has these variants will have a lower energy for the entry of SARS-CoV, and it may be related to the resistance or susceptibility to the SARS-CoV. And we also analyzed the SARS-CoV test results from the UK Biobank data. Over 53% of individuals are tested for the diagnosis of COVID-19, And we can find that this variant may be related with the SARS-CoV positive or infections. But as you can see, the wide range of odds ratio and CI levels, it has a lack of statistical evidence yet. And for the deeper understanding of the molecular mechanisms by which mutated residues affect the binding affinity, we analyzed the MD simulation trajectories, and finally we can conclude that the structural changes due to the mutation would play a pivotal role in the binding affinity with SARS-CoV. We also acknowledge the limitation of this study. First, we only had limited positive cases, but it is still interesting to us, so we reported this result. And second one is the effect of glycan attached in the surface of S proteins were not considered. And by the way, the contribution is still clear. We prompt confirmation and rapid evolution of SARS-CoV is available, and we add our understanding for the effect of the mutation of ACE2 for the SARS infections. And the related paper was accepted at PLOS Computational Biology in July, and you can find more details of our study. Thank you for your listening, and please note any kinds of questions for this one. Thank you.